My name is Janine Scholefield. I'm going to talk to you today about a little project we've been working on for the last few years called Finding NEMO, and how we have used super resolution to, in essence, diagnose disease in a dish. But before I launch into the talk proper, I think it's important to place the work that I'm going to talk to you about in the context of the research that our lab does. And I think perhaps one of the best ways to explain that is to quote Louis Pasteur, someone that my boss regularly reminds us of, where he said that the pathogen is nothing, the terrain is everything. He wasn't alive to see Prof. Morris's beautiful work, but the terrain for us is the host cell. And so part of our research is trying to better understand our host cell machinery so that we can better target or impede or prevent pathogens from hijacking our host cell machinery to their own advantage. So I'm going to go through a couple of basic concepts, and forgive me, I think the audience here is fairly au fait with them, but it's important to start at the beginning. And what I want to show you here is not really what a cell is. I suppose when I was, matric biology or undergrad, I kind of had this idea that a cell was sort of this like fluid-filled sac with the DNA localized in the nucleus. But more importantly, what I want to get across here, I think the concept of what a cell is actually how very, very small it is. And there are two reasons why I want to show you that. Not only for us to get the concept of that, but also so that you can get an idea of the type of microscopy that we're able to do in our lab and the very, very high resolution we're able to achieve. So this is a really great animation taken from the University of Utah. So, and if we imagine that we start off with something that we can all visualize, a coffee bean, and we start magnifying in from that, we very soon, but at now micron level, start seeing your amoeba. And there we go, as we get closer and closer, there's one of the first cells that are really useful to us, a skin cell. And there's your fluid-filled sac with your nucleus in the middle, and skin cells are something that we work a lot with. They're very handy for visualizing, for doing imaging. But that's at the 30 micron stage. And at 30 microns, almost most bog standard microscopes can pick that up. It's really not too much of a problem with your average fluorescent microscope. But we're interested in seeing what's actually going on inside. We want to understand that terrain better, so we need to understand the machinery better, so we need to get closer. If you are lucky and you've got some pretty rad microscopes, you can start getting, there's your bacteria, which are fairly easy, and there's your virus, well, a rather large, please don't freeze. There we go. Okay, so a confocal microscope will get you to visualizing these, to resolve these at an almost individual level. The microscope that we have in our lab, which is, to the best of my knowledge, the only one in the country, gets us down to this level here. Okay, so ribosome, almost antibody level. So we can resolve down to 20 nanometers, which is 10 times more than a confocal microscope. Let's get back to this. So now that we've got an idea of cell size and what our microscopes can do, let's take another look at another concept that we're particularly interested in, and that is the signaling pathway. Every cell in our body, by and large, and I'm making quite a large generalization in the way that I'm talking about a signaling pathway, but every cell in our body must have an innate ability to respond to an attack of some sort, a stimulus of some kind. And here I've drawn them clearly not to scale as a virus and a bacteria. And what happens with the signaling cascade is the cell recognizes something that's not supposed to be there and creates an appropriate response that can then lead to ultimately either the release of some kind of other stimulus, a cytokine to call other cells in, or stopping that particular attack. In essence, that's what a signaling cascade is. So it's this idea that you've got proteins activating one after the other in series. And if our lab is interested in what's going on in the cellular machinery, we need to look closely at how that signaling cascade is controlled. So if we imagine that, as I said, I keep referring to this sort of fluid-filled sac of sort of proteins swimming around in the cell and activating one after the other, it's actually not at all like that. And this is probably the best way to look at it. Moving from the outside of the cell through the cytoplasm all the way through to the nucleus, you can see extensive architectural functionality. And for me, it's almost akin to a city, going from the outside, moving through, and then to the headquarters here. There are rules about how proteins can be activated, how signals can move through, and a lot of those rules are dictated by the structure, the higher order structures of those particular proteins, whether they be full organelles or even the cytoskeletons of the cell. So what is a higher order structure? Very simply, it is a multimeric complex made up of many base units. It's akin to Lego blocks. As long as you follow the basic rules for how they would be joined together, you could create kind of almost any shape that you wanted of an open-ended structure. And there are examples of these in biology, and I alluded to one earlier on. The cytoskeleton is a classic example. There are also examples in DNA, which I'm not going to get into now, but are becoming increasingly interesting. But why are they important in biology? So I'm going to bring up a couple of concepts here, and just bear with me. One is proximity-enhanced response. So what I mean by that is that if you can imagine that you have a whole lot of proteins, rather than being spread out across a cell evenly, that are actually localized physically in physical proximity to each other, there is an idea that if one recognizes a signal, is pinged, that it can very quickly activate the rest. So instead of waiting for the signal to slowly permeate through the cell, you have an enhanced response which is very fast, very rapid. But at the same time, if your protein isn't completely spread across the cell like this, you can imagine that you have limited numbers of these structures dotted across around the cell. And so what you land up with is a threshold response, because there will be parts of the cell that might sense a signal, but won't respond because those higher order structures aren't there. It has to be a true stimulus, a full attack. And this way the cell can prevent it responding from false alarms. And what that means is that you get a reduction in biological noise. So as a result of all of these and the spatial control of signaling, what does this all mean? What kind of system would you create? Well, it creates, ironically in biology, a binary switch. It creates an on-off switch. And one of the best ways to visualize this is that, I suppose, instead of having sort of a dose-dependent reaction to an increasing amount of stimulus and then response, you get a delay. So you get a delay in noise filtering. And what the cell's now able to do is regulate something where it says, I'm not going to respond to a false alarm, but when there really is something there, I'm going to switch it on as quickly as possible, which is actually fairly remarkable for a biological system. So if we want to find higher order structures, how do we do that? Well, that's where super resolution microscopy comes in. If we can imagine that we have a structure that looks something like this, and that's the kind of scale we're looking at, about 250 nanometers, each one of these individual molecules makes up the entire structure. And if we wanted to fluorescently, sorry, I'm just losing my mouse. Where's it gone? Fluorescently label those, unfortunately, because of the physical properties of light, what you're going to land up with is something like that. We can't actually break through that diffraction limit of light, which is 200 nanometers, okay? Even though the whole structure is larger, the individual molecules that make up this delicate structure are much smaller. So that's what you would land up seeing with even with a confocal microscope. But a couple of years ago, almost 10 years ago, a number of scientists who actually won the Nobel Prize last year in chemistry figured out a way to cheat light, to trick it. And what they did was they came up with two very kind of basic rules. The one is that, well, if we know what the diffraction limit of light is, and we can find the center point of that one bright spot, then we can pinpoint and assign a center point to that. So we can actually say, right, even though there's a big blur, that is the molecule, it must be positioned there. The problem is that if you look at this particular structure, you can't actually distinguish between the blobs. So they came up with a second way of tricking light. And they said, what we're going to do is we're going to switch on each molecule at randomly. So they're not all on at the same time. And by combining these two techniques, you land up after reconstruction with a structure that actually looks pretty damn close to what actually exists. So I want to just show you a little bit of what this looks like, and I hope this is going to work. And this was taken by our good friend and colleague of ours, Ricardo, who actually started this project. And he'd been working on the microscope for hours and hours when he was based at Pasteur. And he went to go and sit on the steps overlooking the Eiffel Tower, and he suddenly realized that this would be a classic way to show how what we actually see under the microscope, what the camera is then pulling in, and then post what the algorithm does to the reconstruction. So you're going to see the flashing lights of the Eiffel Tower, what the camera then is able to pinpoint, and what the reconstruction looks like. So what I hope you can garner from this is that though we see some detail here when we're actually looking at it, you get much more exquisite resolution post-reconstruction. And that's basically how super resolution microscopy works in the example that we do. So let's get back to NEMO. What is NEMO? Very simply, in that signaling pathway, and in fact probably one of the most important signaling pathways in the cell, the NF-kappa B pathway, NEMO acts as one of those critical guardians. It's the critical regulator in leading to a response to a stimulus. So if that's its job, it became obvious to my boss, Musum Schlanger, and his colleagues in Pasteur that this would be a good candidate for having a higher order structure. But if that's going to happen, we need to understand the rules. Remember, akin to the bricks, the Lego bricks, in terms of how you join them together, you must follow some basic rules on how they are joined. So this is what NEMO looks like. We've got some basic ideas. It's a coiled dimer, so you can see two NEMOs wrapped around each other. We also know that NEMO is joined at the one end by a protein called IKK beta, and at the other end, we know that it is linked non-covalently joined by polyubiquitin chains. Now, there is information that suggests that these polyubiquitin chains could bind another NEMO dimer. So that's one join, that's one rule of binding that we can follow. There is a very small amount of information which also suggests that these IKK beta subunits could actually bind each other. So now we have two rules, how we could join this NEMO dimer base unit end to end. So if you play around in Adobe Illustrator a little bit, you can start joining them together based on those rules. And you can see that you can start very easily creating, hypothetically, a lattice structure of open-ended size. What's important, and this is critical, though this lattice could extend in microns, which is above the diffraction limit of light, because it is held together by these very delicate bridges that are below the diffraction limit of light, the only way we could possibly visualize this is with super resolution. So let's have a look and see what NEMO looks like in resting cells. I'm afraid the red's gone a little bit squiffy, but fortunately, actually, you're not really supposed to see any structure in these particular images that we've taken. So this is using conventional microscopy. There's the nucleus of your cell, there's the outside of a cell, and we've stained NEMO in red. And as you zoom in and magnify those images, there's really no information there that you can tell. If we apply super resolution microscopy to a similar cell, we see this. So again, at the same magnification as here, you can very easily start to see these higher order lattice structures that exist compared to what we would see with a conventional microscope. And you can see that they do indeed extend almost over 1.5 microns. But because they're held together by those delicate bridges, it's only because of super resolution that we can see them. Because we're scientists, we can't just check that we're doing this in one cell type, so we had a look in several other cell types. And again, this is what they look like in skin cells, what NEMO looks like in skin cells. And again, these lattice structures we can find in multiple cell types, and you can see that, in fact, this one I couldn't even fit in that one particular magnified image. So they are enormous. But now we know that they exist. We have to find information about what holds this lattice together. So we did a lot of experiments, but I'm just going to show you two, where we very specifically prevented that ubiquitin chain from binding to NEMO. And when you look at conventional microscopy, we can't see that that's done anything to the NEMO protein staining at all. But upon super resolution, you can see that we've all but ablated those structures compared to what they look like in untreated cells. So now we know one rule. We know that we were right. We know that the ubiquitin chains are holding these things together. We did a second experiment on the other side where we inhibited the IKK beta binding. And what you can see here is compared to cells that make this beautiful lattice structure, when we remove IKK, that lattice structure is destroyed. And we were able to see that in a second experiment when we did dual super resolution, so looking at two proteins at the same time. And basically what I want you to take from this is that the IKK beta and NEMO are actually in physical close proximity to each other, whereas another arbitrary protein that's not involved in the immune response doesn't seem to co-localize with this lattice at all. So now we know the rules for how this lattice is joined together. That's great. But we've been looking at cells in a resting state. We haven't added a stimulus. These lattices pre-exist, which kind of makes sense because you want the response to be ready as quickly as possible. But what would actually happen if we added a stimulus upon super resolution microscopy? So if we look at conventional microscopy, you can see that in an untreated cell, NEMO has this sort of speckled appearance here, but when we add a stimulus, it snaps into these really tight punctate NEMO aggregates, okay? So we can see that with conventional microscopy. What happens when we look at super resolution? So I'm going to just zoom into this particular box here, and what you should be able to, I'm sorry, it's a little difficult here, but what you should be able to see with box A, there is a large punctate structure. That's activated NEMO. That's what it looks like upon super resolution and merged. But the cell still has some of these preformed lattice-like structures. There's no information here with conventional microscopy, but they're here in super resolution. And what we think is happening, you may recall I mentioned the term proximity-enhanced response. This is some of the first evidence to suggest that that might actually be happening, because it occurs to us that these larger lattice structures, if one is pinged, one of those NEMO molecules is pinged, it's almost as if they are folding in on each other and activating all of the other NEMO molecules so you get a rapid response. And that's what we think these aggregates are, in fact, doing. So one of the last pieces of data that I just want to show you takes us away from the actual basic research, but more towards a clinical phenotype. So we were very fortunate to work with collaborators at Pasteur, and they supplied us with skin cells from individuals who had a healthy NEMO gene compared to skin cells from individuals who had a mutated NEMO gene. If we look at conventional microscopy, again, the healthy cells are able, once stimulated, to form those punctate structures. So that immune system, that NF-kappa B pathway is working. But when we look at the patients, you can see that there's no difference. They can't form those structures. But it was unclear to us as to why, because these patients still have NEMO protein. It's a very, very small mutation. So what we wanted to do was then look at their resting cells to see if super resolution could give us an idea of what had actually gone wrong. And indeed, that's what we found. We realized for the first time that the reason why these patients aren't able to mount that response is because they don't actually have the preformed lattice. That open-ended structure isn't there. So they can't snap into those NEMO-induced aggregates compared to what a healthy cells from a healthy individual look like. So to summarize, what you've got here is that in an untreated or healthy cell, we have these massive NEMO, open-ended NEMO lattice structures that can extend microns in distance, but held together by very, very thin bridges. In a stimulated cell, you get compact NEMO aggregates, almost as if those NEMO lattices have folded in together to activate a rapid response. But in cells that lack polyubiquitin chains, IKK beta dimers, or mutations in NEMO, they don't have a preformed lattice and can't mount a response to any kind of stimulus. So why is this important? It's the first visual evidence of a higher order structure in the immune response, to the best of our knowledge. It provides a mechanism for a binary switch to control the immune response, advancing our knowledge of our cellular machinery. And it shows, I think, how super resolution microscopy could actually be used to diagnose disease in a dish. With that, I just want to thank our collaborators, obviously specifically I'm in Musa Mhlanga's lab. Anka Savalescu was involved in this particular work in our lab as well, and our collaborators Ricardo and Fabrice overseas. And with that, I'll stop.